Click for IB Biology. Here we're looking at further regulation of gene expression, one in which we have a hormonal impact on gene expression, which we're going to look at cortisol. And the second example we're going to look at affecting gene regulation is a biochemical impact. And for that, we're going to look at the availability of lactose, sugar in milk, and the Lac Operon model. So, how can a hormone affect the regulation and expression of a gene? Stress in humans is detected by the hypothalamic region of the brain, which is approximately here, and results in the secretion from the pituitary of a hormone called ACTH. This travels through the bloodstream, and its target tissue is the adrenal glands on top of the kidney. The adrenal glands in turn produce the hormone cortisol. I'll use C to represent cortisol. And cortisol travels through the body and will then pass through the cell membrane. And of course, as this is a steroidal hormone, the membrane presents no resistance to the cortisol. It can pass straight through the hydrophobic layer. Their cortisol is picked up by a receptor molecule, which is called a glucocorticoid hormone, a glucocorticoid receptor. And we'll just abbreviate that to GR and think of that as a receptor. Now together, the cortisol and the receptor are able to bring about a change in the expression of genes from in the nucleus, and those genes convert the stored carbohydrate glycogen. It breaks it down and makes glucose, so that's glucogenesis is the name of that process. So enzymes are produced that break down the insoluble polymer glycogen and produces glucose, and the glucose is dumped into the bloodstream as a response to the stress, no doubt to provide molecules, energy molecules for respiration in different parts of the body. At the same time, however, that's the positive impact. The negative impact of cortisol is that by a different mechanism and pattern, it can result in inflammation in the tissues of the body, and of course that has a negative detrimental effect on health. So the hormone regulating gene expression is cortisol, and it brings about the production of glucose and inflammation through changing gene expression in cells. If we look for a biochemical signal to regulate gene expression, then the one that we're going to look at here is the regulation of genes which break down the milk sugar lactose. Now in human beings we would be looking at lactase, but in bacterial cells such as E. coli, the enzyme which breaks down the lactose is this one here, is galactosidase, and produces glucose and galactose. So within milk there is within milk there is E. coli, and they have the ability to break down milk sugar, lactose, into glucose and galactose, clearly for respiration. However, and this is a very, very important key point, E. coli biochemically have a preference for glucose, so they won't do this if there's a high concentration of glucose present. And this is called Catabolite repression. This mechanism was determined by Jacob and Monod in 1965, and for this they gained the Nobel Prize in their publication of what is known as the Lac Operon model. So what we're trying to do is we're trying to explain how the biochemical signal of glucose or lactose changes gene expression. Now, when we look at the gene locations, then of course, let's not forget E. coli has a nucleoid, and on that nucleoid there's a region in which we have three enzymes, Lac Z, Lac Y, and Lac A. Now, Lac Z is responsible for the enzyme. I'll put E to remind us. Lac Y is responsible for producing a permease. In other words, it allows the glucose and the galactose to be taken through the cell membrane. And Lac A is involved in a kind of filtering function to protect the E. coli cells when it's absorbing these products from molecules which might do it harm. So in the presence of lactose, we're expecting lactose permease to allow lactose to be taken into the enzyme. Lac Z will be expressed and bring about this catabolism. But remember that E. coli prefers glucose to lactose. And so this is where our biochemical signal comes into operation. So let me try and lead you through the mechanism of this variable mechanism of response. So we have here our Lac Z, Lac Y, and Lac A genes, and we've already been through what they produce. Upstream of these three genes, now I should have said that where we are looking at three genes being controlled together, this is what an operon, that's what an operon is. So that's why it's called Lac, I should put it there, Lac Operon. So Lac refers to the lactose, and operon is the fact that there are three genes being regulated at the same time. So if we just go through the mechanism, we've got the three genes, we have an operator region here, we have the promoter region here, to which the RNA polymerase will attach, and then upstream of that again there's a so-called cap region. Now, under the influence of high glucose levels, what happens is that the cap overlaps the promoter region and prevents the binding of polymerase. This means that the operon does not become active and the lactose is not digested or absorbed into the bacterial cell. So let's think about our bacterial cell here. So if it's surrounded by glucose and lactose, but the high concentration of glucose prevents the polymerase from bringing about the expression of the gene, and so glucose is used in preference to lactose. That's our biochemical signal. In low glucose level, the cap is removed and the promoter is free to accept the RNA polymerase. The RNA polymerase will bring about the expression of Z, Y, and A, so that, let's draw our bacterial cell. So if there's only low levels of glucose but large levels of lactose, then Y will have the lactose taken in, A will make sure that it's only lactose, and then the Z will bring about the enzyme to digest the lactose. So the bacteria can switch from either using glucose or lactose, but has a preference for glucose. The addition to this is that the high lactose levels, very high lactose levels, also can remove what's called a repressor protein, and this enhances the expression of these genes here. and so the lactose is metabolized more efficiently. So this is an example, a variable example of biochemical signals switching on either the metabolism of glucose or the metabolism of lactose, with a preference for glucose included. Jakob Monod, Nobel Prize for this, 1965, I think it was. Here's the link to the channel should you wish to subscribe, and to the playlist to take another look at the videos in this topic.